Migraines are more than just headaches. They are unwelcome guests that can disrupt our lives unexpectedly. They are not just simple headaches but complex neurological events that impact a significant portion of the global population. Migraines manifest as severe pulsating headaches accompanied by additional symptoms like nausea and heightened sensitivity to light and sound, which can last for varying duration. Familial hemiplegic migraine or FHM is described as a rare autosomal dominant subtype of migraine with aura. FHM is characterized by aura symptoms that include motor weakness and paralysis, often unilateral. Individuals with FHM have an increased risk of experiencing typical migraine attacks compared to the general population, suggesting a potential overlap in the pathogenic mechanisms between FHM and typical migraine.
Familial hemiplegic migraine type 1, rare neurological disorder that offers intriguing insights in both genetics and migraine pathophysiology. FHM1 is primarily caused by mutation in specific genes, notably the CACNA1A gene encoding the A1A subunit of voltage-gated calcium channels. This mutation disrupts calcium ion influx into neurons, leading to aberrant neurotransmitter release and neuronal excitability, ultimately triggering migraine attacks. The pathophysiology of FHM1 involves complex interactions between neurons, glial cells, and vascular components within the brain. One feature is cortical spreading depression, a wave of neuronal depolarization followed by prolonged neuronal suppression, thought to underline the aura phase of migraine. FHM1 is a rare condition, estimated to affect approximately 0.01% of the population. It follows an autosomal dominant pattern of inheritance, meaning that individuals with a single copy of the mutated gene have a 50% chance of developing the condition. Living with FHM1 can be challenging, as often leading to debilitating migraine attacks, hemiparesis, and other neurological symptoms.
Familial hemiplegic migraine 2 is a genetic disorder characterized by mutations in the ATP1A2 gene. It was first identified year 2003 in two Italian families with mutations in their ATP1A2 gene. It only affects a small amount of the population and is considered relatively rare. Membrane potential, cell volume, and neurotransmitter uptakes in cells are all disrupted by defective sodium-potassium pumps induced from mutations in ATP1A2. This can cause neurological deficits like seizures and epilepsy in FHM2 patients. Impaired ion gradients may also affect neurotransmitter uptake and clearance, affecting synaptic transmission and excitability. FHM2 or familial hemiplegic migraine 2 highlights the crucial role of the sodium-potassium pumps in maintaining neuronal functions and synaptic transmission.
The gene for FHM3 was identified in 2005 through genetic studies in three German families. A mutation in SCNIA, the gene that encodes the alpha subunit of the voltage-gated sodium channel NAV1.1, was found to be associated with familial hemiplegic migraine type 3. This discovery provided insights into the genetic basis of this specific form of migraine. NAV1.1 channels are predominantly expressed in the central nervous system during late postnatal stages, particularly from the second postnatal week onwards. While they are a minor component of total brain NAV channels, NAV1.1 channels are broadly distributed in various types of neurons. These include hippocampal and cortical pyramidal cells, as well as inhibitory interneurons. These channels are specifically localized in the soma and proximal dendrites, suggesting a crucial role in the dendritic excitability and synaptic signal processing. The FHM3 mutation was identified in SCNIA affects the cytoplasmic segment linking domains 3 and 4 of the NAV1.1 channel. This mutation has been shown to cause accelerated recovery from fast inactivation of recombinant human NAV1.5 channels expressed in TSA201 cells.
In order to manage the effects of familial hemiplegic migraine, there are drugs that aim to regulate the continuous occurrence of cortical spreading depression and even the impulsive activation of the cortex. Based on the journal, valproate, topiramate, amitriptyline, propranolol, and metisergide are the primary medications for the management of episodic migraines and even controlling seizures, specifically valproate and topiramate. Alongside this is the continuous search for the potential drugs such as tonabersa, which is under numerous trials to make sure that it is 100% effective in regulating the continuous occurrence of cortical spreading depression within the brain.